Good morning, everyone. I'm Zoya Gluzman-Poltorak, CEO of Forta Bio, and I'm very happy to present to you our actual debut presentation as a new company. We're developing novel class of targeted oligo for autoimmune and oncology. And so my personal background, actually, therapeutic development for over 25 years in the space of cell and gene therapy. And being in this space for so long, I know all the limitations and complexity and all the organizational and operational issues that can come from the cell and gene therapy. So this is why in the last year, I reposed and just wanted to understand what the next modality I would love to develop. And Forta Bio actually came to address critical gaps in genetic medicines. So we picked their validated mechanism of action with a targeted delivery, high specificity and the results of high safety, and simple manufacturing. And this is really our focus, simple manufacturing of oligo with a broad application. So for the mechanism of action of Forta Bio is targeted telomere disruption using modified nucleotide. This 6-tiodeoxyguanosine is packaged in 5 to 6 molecules together and attached to the targeted domain. And this targeted domain actually leading to specific targets. This company was founded in 2025 this year by me, my business partner, Valeria Pavlotsky, and based on the foundational technology from City of Hope by Professor Marcin Kartilewski. So telomerase as a target is clinically validated, and it was approved, the first product was approved last year. The first product with their high specificity in TERT positive cells, but unfortunately in also high toxicity. And this high toxicity on target but off tumor toxicity actually limit broad application of telomerase inhibition. What Forta Bio is bringing is dual specificity and dual mechanism of action. By selective delivery to specific receptors, and the first generation is a scavenger receptor we're targeting through their CpG domain. We can specifically target pathogenic cells that express scavenger receptor, and this is B cells, myeloid cells in the first example. And scavenger receptor is a family of receptors, so it cannot get to antigen escape the same way as antibodies or even CAR-T with their mono or even dual specificity. So we can target this through scavenger receptor, and it will kill only the cells that express TERT. So you can see, for example, in the B cells, when we target their B cell that proliferative lymphoma B cells, they expressed very high level of TERT. It will kill directly the cells, expose the DNA, and activate antigen-presenting cells. It also can go to myeloid cells, but they're fully maturated and they don't express TERT. This is why there is no killing effect on dendritic cells. But we can do modulation of the dendritic cells, and depends on how we structure our oligo, we can either activate or inhibit activity of their of dendritic cells. So we have really broad platform. Our platform includes three parts. The first one is Forta-Rec that we can use for autoimmune disease, Forta-Onc that we can use for oncology, and Forta-Apt, when we use aptamer for targeted delivery, we can target actually any kind of cells. And with the Forta-Rec, we specifically deplete B cells, but also downregulate dendritic cells through inhibition of dendritic toll-like receptor. In the Forta-Onc, we activate actually dendritic cells, and through toll-like receptor, we increase the immune activation. with the Forta app, and we have the proof of concept for the Forta app for glioma and other targets. We can target 60 go to any potential target that we can imagine. So in addition to this, we have extensive collaboration with the aptamer expert that can bring and develop libraries of 10 to the 12 aptamers, and we can screen very efficiently using robot. All this amount of data will feed our AI model, and we can select specific targets. That model was already validated in terms of developing aptamers. We have a very strong candidate for glioma, glioblastoma. We just submitted SBIR for IV injection and treatment glioma and glioblastoma using this aptamer. We have additional candidates for lung and also bone marrow, and potentially we can develop any candidates for using this methodology. So just to emphasize, the advantages for Forta Bio platform is the dual selectivity and dual specificity with very selective and specific elimination of the pathogenic cells and activation immune system. On top of this, we have superior safety and very easy manufacturing compared to other modalities. And the comparison to all the existing telomerase inhibition's main point actually really their targeting delivery. When we do targeting delivery, we preserve immunity, we preserve T cells, we preserve hematopoietic stem cells, and this way safer modality, and also we can use, of course, because of the safety, we target pediatric population that wasn't really possible with other telomerase inhibitors. So this is the results showing that many cancer cells that express scavenger receptor. And we have done screen on all these hematological and solid tumors and show very extreme high level of efficacy on all these tumors. So we have really broad application in terms of where we plan to go. Here, the publication was just recently published in July 2025. We showed that it's a very specific targeting of B cells, dose-dependent targeting of B cells. This also goes to dendritic cells and B cells, but completely elimination of the targeting of T cells. So we preserve the T cells, and it's very important in terms of oncology, population, and autoimmune. On the next slide, you can see how important the immune system and long-term immune response. So when we treat immunodeficient mice, the effect is not as prominent as when we treat immunocompetent mice. And you can also appreciate the comparison to single nucleotide, modified nucleoside, that there is no effect, there are effect not as complete as in our case. So long-term effect, this is really the focus for us, because we, in opposite to chemotherapy or even antibodies, we really want to rely on the long-term effect of immune system. And it's some kind of similar to CAR-T, but way more controlled and easy to manufacture. So in terms of long-term effect, we have very prolonged survival. Now we have the new data set with the new candidates up to 70-day and longer survival compared to 100% mortality in the control group. And the results really are exciting and prominent, and the importance of the T cell preservation and T cell response. You can see how the T cells, all the population, CD4, CD8, and Tregs preserved and active. And this is what actually kicking long-term immunity. The biodistribution show very broad, but also very fast elimination to multiple cell population. However, you can appreciate the DNA damage is very specific, only in tumor cells. So it goes to dendritic cells and other cells, but the death, actually the killing effect, only in the tumor B cells. We also did very extensive safety profiling of Forta-ONC-60 in humanized model. using hematopoietic stem cell, human hematopoietic stem cell humanized model. And after 6 injections, very intensive regimen and pretty high dose, significantly higher than we need for efficacy, we don't see any effect on hematopoietic stem cells or T cells or myeloid cells. There is some partial depletion of B cells, but it's completely non-clinically significant, and there is no effect on bone marrow or any other organs. The PK is pretty short. It's eliminated through kidney, and this why kidney is really our good target, really fast, within 1 hour, but we can control this using a prolonged IV injection, and this can be actually used in the clinical setting. Additional point that there we really preserve the functionality and differentiation of hematopoietic stem cell, and here is just one example, but we have multiple examples of differentiation preservation. We have extensive IP, we license out IP from City of Hope, but we also work with our attorney Wilson Sonsini on our new Forta Bio IP. This is the pipeline. We focus on Forta Onc. It's most advanced in terms of development and ready for IND enabling studies, both for hematological malignancies, but also for solid tumors. And under solid tumors, we can do multiple types, and mainly prostate, kidney, as I mentioned, very good target for us, but other solid tumors also good candidates, and we can do blanket design. We also have the Poltorak for autoimmune, as I mentioned, nephropathies, probably really also good target for us. And Forta Up, glioma, glioblastoma, we just submitted SBIR, and we have really good candidate. So where we are now, we just founded in 2025. And on strong foundation from City of Hope, they just for this project, they are in Marcin's lab. They have $1.7 million of this DOD and NIH grants. And this actually was built on many, many years, over 15 years of research and additional grants in area of oligonucleotide development. We have really strong IP exclusively licensed out from City of Hope, and we work on additional IP. We have multiple SBIR grants currently in the submission. Collaboration already well developed in terms of IND enabling and clinical development, and we will use orphan designation as a tool for expedited delivery. This is their plan for their seed money. We're currently fundraising. Timeline, 2 years in IND and clinical enabling, and we actually can go either hematological malignancies or solid tumors. It really depends on their fundraising and partnership. We're actively working also on the partnership with the big pharma and collaborations. In the summary, this really big opportunity for novel class of targeted oligos with very defined mechanism of action and high efficacy and high safety profile. We have 3 proprietary platforms. This platform's very diverse and very modular and very easy to manufacture. It's clinically validated in terms of target. This strategy, orphan first, actually will help us to expedite development and get to their approval really fast. And as I mentioned, within 24 months, we plan to be in the clinic and with multiple returns and collaborations. Thank you for your time, and I'm ready here for questions.
Thank you, Forta Bio.